Thank you for joining us today. I'm excited to explain our technology to all of you and mostly its applications. We do things a little bit differently. We're a whole genome imaging platform. We don't sequence, we don't read the A, C, Gs, and Ts. And I'll hope to convince you that that's actually a real good approach. But what we're really trying to do is call structural variation genome-wide, and we do that better than anyone else. When I'm talking about structural variation, I'm talking about the unbalanced events, deletions, duplications, repeat expansions that are pretty easy to pick up with some other technologies, but also the balanced events like translocations and inversions. So the standard of care right now to detect structural variation genome-wide is still based on a method that's half a century old, where you look at patterns on intact DNA molecules using some form of microscopy. And when NGS came along, there was this promise that all of this could be replaced by short-read sequencing if we simply could read every letter of the genome, and then we would know all the structural variants. But that didn't work out. And we think that didn't work out because the NGS crowd didn't bring a microscope. They brought this. And this may seem silly, but I would challenge you to tell me how many pieces of strawberry are in this bowl and where they are after I turn this thing on. And that's exactly the first thing that you do when you run an NGS run on a sample. You take a perfectly intact, beautiful genome and you chop it up into millions of pieces. And then you try to make sense of all these pieces to figure out what it looked like in the first place. That doesn't make sense. The other issue with short-read sequencing is that it has a big problem handling the repeats of the genome, and just about a third of a human genome is unique, and 2/3 consist of repeats. And the majority of the single repeat units that you see on the slide are already longer than the read length of short-read sequencing. So it's almost impossible to make sense of those, and especially if you have long tandem arrays of these repeats occurring at multiple sites in the genome. And that's why cytogeneticists still study structural variation by looking at intact patterns on DNA. And that's what we do at Bionano as well. Except for we add this nanochannel array, which has hundreds of thousands of channels that fit a single DNA molecule, linearize it and image it. So you get pictures like this of the genome instead of just a few of these chromosomes. And our microscope is called Saphyr, and it automates this process for you. Now, Bionano has been around for a while. This is our first time that we are here at AMP, and why? Why are we here now? Well, we can call these structural variants better than anyone else because we have the longest molecules. We image molecules that are on average more than 350 kb. That is orders of magnitude more than with long-read sequencing and, of course, short-read sequencing. Our instrument gives you 400x coverage of a human genome, and that allows you to find structural variants down at 5% variant allele fraction. I'll show you this in more detail. All of this costs just $500 right now, and we're on a path to bring that down. The instrument is high throughput. It can handle 3 to 9 human genomes a day, depending on how much coverage you want. And of course, we can get you as many of these instruments as you need. And sample to data is somewhere between 3 and 5 days, depending on how much coverage that you need. Now, let me walk you through the workflow real quick, and then we can talk about the applications. Bionano produces a Saphyr instrument, which images the DNA. We have the chips for linearization of the molecules. We have kits for DNA extraction and labeling, and then software that analyzes the structural variants for you. Now, if you want to image megabase-sized DNA, the first thing that you need is megabase-sized DNA, and that's not obvious because any standard method to isolate DNA, whether it uses spin columns or magnetic beads or simply precipitates the DNA, exerts so much mechanical forces on the DNA that these molecules break. The molecules break in fragments that are maybe 30 kb, 50 kb. That's considered high molecular weight DNA. And that's just not enough. We want megabases. So we developed this method with a partner where we lyse the cells in solution and then grab onto them with a disc, a silica disc that has a 3D structure that can hold onto micrograms of DNA. And then you simply wash and elute in a clean tube, and you have megabase-sized molecules, massive molecules that just have not been exposed to any mechanical forces, and so they stay intact. Our sample prep method takes just 4 hours and is automatable. We're working with several automation partners right now. We can use fresh or frozen blood as input material, less than 650 microliters, and bone marrow aspirates as well. We can work with 1 million to 1.5 million cultured cells, or 10 milligrams of fresh or frozen tissue. We cannot work with FFPE samples because the DNA is simply too damaged, too fragmented to get long read information out of that. So here you see the distribution of the molecules that we image on a good but not exceptional run. We see the N50, which represents that half of the genome is imaged in molecules larger than 434 kb, just massive here. And we have molecules that tail off all the way to 2 megabases and more. Again, orders of magnitude, and even long-read sequencing gives you PacBio, Oxford Nanopore really fall right at the start of our graph here with our average read length. Okay, we have megabase-sized DNA. Next, we linearize that, we label that DNA in a method that we call direct label and stain. Staining is the backbone. We stain that with an intercalating dye that gives you a nice blue signal that tells us where the molecules are. And we label a single 6 base pair sequence motif that occurs all throughout the genome. We place 500,000 of these labels all throughout the genome. And again, no probes, just a single enzymatic reaction, highly efficient, and it labels the DNA all throughout. And now you get something like this, megabase-sized DNA with a specific green fluorophore attached at specific sequence motifs. And then we linearize all of this into our nanochannel arrays. These channels fit a single molecule of double-stranded DNA and stretch it out uniformly. And because of that uniform stretch, we can collect all the data that we need just by taking a picture. That picture tells us the distance between these labels, and we use that distance to call structural variants. It looks like this, and it's easy to see how each of these long molecules, megabase-sized molecules, sit in each of these channels on top. Our instrument uses electrophoresis to move molecules in, image them, move new molecules in, image them over and over and over again. And with that, we collect the 400x that I mentioned in 3 human genomes in parallel. Our spec says 48 hours, but in reality, we see that in about half of that time. And if you use constitutional cases, you don't need to collect 400x. You get by with 100x for highly sensitive structural variation calling. And we can do 6, next few months, 9 human genomes per 24 hours. The instrument has 2 chips for continuous running, so once the data is collected, it'll move over to the next chip, and you can replace that first one, and each chip can hold 3 human genomes. Now we're imaging a massive amount of data out of these chips, and then our software will convert these images into digital representations of the molecules. And out of these digital representations, we do a pairwise alignment of every molecule that we imaged with every molecule that we imaged. What that means is that we built a genome completely de novo, and we built a genome that is really there. Our software is not aware of the fact that it's assembling a tomato genome, or a standard human genome, or a highly rearranged cancer genome. We built all the variants that we see, again, completely de novo. And out of these alignments, we built consensus genome maps that represent the average spacing of the molecules that align with that map. These consensus genome maps are then used to call structural variants. We can compare samples with each other, tumor normal, for example, or a trio analysis for genetic disease, or a cell line with an engineered version of that cell line. But a simple analysis relative to the reference could look like this. If this is HD19 or 38, we import that sequence, and we know what the sequence is, so we know where these label sites are supposed to be. We align that to our de novo maps, and you see that all these patterns align perfectly except for right here, so that would be a deletion. And our software detects that automatically genome-wide. And we can do that for every type of structural variant. Deletions starting at 500 base pairs, as I mentioned, are called by the distance between 2 labels decreasing. An insertion is detected when the distance between 2 labels increases. We call that automatically genome-wide at 500 base pairs as well. I'll show you some examples of repeat arrays. We can call those. Tandem duplications starting at 30 kb are detected when a pattern, like you see here, aligns twice, here and here. We detect those direct or inverted starting at 30 kb. A translocation looks like this. A de novo genome map aligns for half with one chromosome and for half with another chromosome, or a different part of the same chromosome. We call those starting at 50 kb. And inversions are detected when a pattern is simply flipped around. So with this map-based analysis, we call every major type of structural variants. Now we have some additional algorithms specifically for cancer. We have a copy number tool that works very similarly to a copy number tool with NGS, or even array if you may. We simply count the number of molecules that align to each part of the genome and normalize that. And based on that, we can call increases and decreases in the copy number profile all the way down to 10% allele fraction. So if just 20% of the cells in your sample have a heterozygous large deletion or duplication, we will pick that up. And then we built an additional pipeline just for those complex heterogeneous cancer samples. We call that the rare variant pipeline. And what this will do is it will grab this 400x of raw data that we collect out of each flow cell and call each major type of structural variants, translocations, inversions, deletions, insertions, and duplications down to 5% variant allele fraction. This is not a panel. There's no amplification. There's no bias. We're not just looking for those variants that we're looking for. We call every translocation genome-wide, anywhere in the genome, down to 5% allele fraction. And you can go down to 1.5% if you simply combine 3 of these flow cells. This is not my most subtle slide, but no one else can do this. No one else comes even close to this type of performance for $500. Now, it's easy for us to say that we're good at this. There's some publications that looked at this, and the first one here compared us against the Illumina sequencing as part of the 1,000 Genomes project, where 154 human samples from 26 different ethnic groups were mapped with Bionano and compared to that same data on these same samples from the 1,000 Genome project. Bionano called 8.5 times more insertions. Insertions are almost impossible to pick up with short read, but even long read sequencing, because these aligners just don't know where to put that extra sequence. We see these insertions on single molecules. We see that these labels move further apart because there's material inserted, and so we're very good at detecting that. And then the Human Genome SV Consortium published fairly recently a comparison between Bionano, Illumina, and PacBio. PacBio is better at picking up some of these variants that Illumina misses, but it still misses most of the large variants. And if you look at this slide here, deletions on the left, insertions on the right, starting from 500 base pairs all the way to 1 mega base pair, you see that this light blue section, which are variants picked up only by Bionano and missed by Illumina and PacBio, take up a massive amount of the deletion and even almost all of the larger insertions. No one else sees these variants. You miss them with any other molecular method. So I want to walk you through a few examples of how this technology is applied in genetic disease first, and then I'll talk about cancer. MD Anderson had a family under their care where 4 children had an extremely rare 1 in a million type of tumor, and applied every known molecular method to this family over the course of almost a decade. Couldn't figure this out. When we imaged the genome of one of the patients, we saw this region of the genome here. You see the molecules that we imaged, and you can even see with your own eyes that there's some repetitive patterns in there. So our software builds a consensus genome map that aligns with those molecules and represents the average spacing. And when you compare that to HD19, you see that this aligns perfectly on the left. Something's going on the right. We built another allele that really aligns perfectly with the reference. It's a wild type allele as well. And what's going on here is that there's a 38 kb segment that's present in 6 tandem copies. This duplication is not present in the controls that are unaffected. What's important also is that this gene is known to be mutated in this pathway in this cancer. So how can we be so sure that this duplication is there? We know this because we have single molecules, quite a few of them, that span the entire 230 kb of that repeat. That repeat is visible. Again, you see these patterns with your own eyes on single molecules. The fact that that pattern is there means that that repeat exists. There's no other answer. There's no other possibility. So we observe structural variants on single molecules as opposed to chopping the genome up in tiny little bits and then trying to make sense of that. That is the power of Bionano. We've had a lot of interest recently from a community studying a disease called FSHD. It's a form of muscular dystrophy that has a very odd mechanism. It's the only known repeat contraction disease where a large repeat subtelomeric on chromosome 4 contracts. If you have the 4qa allele, the pathogenic allele that causes the disease, the non-pathogenic allele doesn't cause the disease, there's another repeat on chromosome 10 that's not involved in the disease. This is very hard to genotype, but with Bionano it's very obvious. You see the contracted allele here, the wild type allele there, and the molecules that align. These patterns at the edge tell you which haplotype is present, the pathogenic or non-pathogenic allele. So based on a number of publications on this disease, we developed some custom software that does all the genotyping for you. It distinguishes the A and the B allele, chromosome 4 from chromosome 10, sizes that repeat, checks another region of the genome that's involved in this disease, and gives you a PDF with all those results. And based on these results, ParkinElmer Genomics announced that they're launching an assay using our technology. This is a big deal for us, and so we're very excited about this. We're building more of those applications for different genes in the near future. So you can think of Bionano as a single workflow in the lab with a menu of interpretations. If you want to do a cancer panel, if you want to do repeat expansions, you can get that data out of this uniform workflow. Cancer. I want to talk about this paper from Jim Broach, who is the director of personalized medicine at Penn State in Hershey, who looked at 12 leukemia genomes and found a number of variants that are really interesting, like this non-Hodgkin's lymphoma case, where the T cells were cultured as the germline control. And the lymphoma cells, he picked up a 3 megabase inversion that cut right through PTEN. PTEN is a known tumor suppressor, and there's dozens of compounds in trials that rescue PTEN mutations. Now, this inversion was not picked up with NGS. It was not seen on a karyotype. Only Bionano could find this, and this could immediately affect the treatment of this patient. And in this paper, he showed a large number of leukemia genes with recurring structural variants. That makes a lot of sense. But also a lot of non-leukemia associated cancer genes with recurring structural events in those 12 leukemia samples. There's an enormous potential there, of course, for biomarker discovery or therapeutic potential. And on top of that, he showed dozens and dozens of genes that are not known to be cancer associated, that again had recurring structural variants in these leukemia genomes. This is just on 12 samples. Imagine what you could get, what you can learn if you look at massive sample sizes on this technology. On top of that, he showed that there was a significant enrichment of cancer genes within 5 megabases of each of these large types of structural variants. So just a massive potential there for discovery. Jim Broach presented a webinar that's available on our website and in print as well. Right here at the exit, when you leave, you can grab a copy of that. And so I think you can see how Bionano can really help you with biomarker identification, compound development and optimization, personalized medicine and patient stratification, but also gene therapy. I don't have a chance to get into that, but of course if you're making CAR T cells or you're doing any type of genome engineering, we can show you any unwanted structural variants that occur in the genome. So I want to finish by talking about a validation study that is ongoing in the Netherlands. Alexander Huizen from Radboud University in Nijmegen and his team are doing a large study based on leukemias, 100 leukemia genome and 50 constitutional cases. And I'm going to skip right through his results slide, which really says here that they believe that this could be a cytogenetics revolution. We of course hope that they are right with that, or we believe that they're right. And that they really see how our technology could replace cytogenetics, because all previously identified variants in these samples from array and FISH and karyotype combined were picked up with Bionano. There's a webinar that is also transcribed of his presentation that's again on the table when you leave. And we have a poster that shows some of this data as well. You can go check it out Friday at 2:45 p.m. Our second poster is on Saturday and looks at metastatic ovarian cancer and ascites compared with each other, matched using our technology. So I'll finish by talking about very quickly about this software workflow that exists where you can integrate whole genome sequence data, if you have that, with Bionano SVs. The problem with Illumina data is that these read pairs are really just too short, and the aligners have a very hard time placing them correctly in the genome. Here you're looking at a 14 kb window and some read pairs aligning as best as they can. Now, Genox built a software solution where you can input the Bionano structural variation calls, and the aligners will use that information to better align these short reads, and now you get single nucleotide breakpoint accuracy for the majority of these structural variation calls. This is a very interesting technology, and if you want to know more about it, come talk to us. And then lastly, there's a method published by one of our customers, not commercially supported by us right now, but to look at DNA methylation. I wanted to mention this real quick here as well. What they're doing here is labeling unmethylated CpGs in green on the same molecules where Bionano applies its standards label in red. So now you can detect structural variants and methylation on single molecules and phase that over hundreds of kb. If you've worked with bisulfite converted DNA, you know that your length of view is very, very short. Now you can look at DNA over hundreds of kb and know that if a promoter is methylated, that a regulatory element 50 kb upstream is methylated as well or not on single molecules. That is all I have for you. There's a survey card on in front of you. If you fill it out, we'll give you a power bank to keep your phones going this conference. We have a booth number 3044, so come see us for the next few days. And with that, I want to thank you for your time. Come see me afterwards if you have any questions about our technology. But I would like to introduce Rashmi Kanagal-Shamana from MD Anderson. He's going to talk about her experiences using our technology in hematological malignancies. Thank you.